I am Paula Sweeten and I am a Research Impact Officer at the University of Glasgow. My job is to work with academic scientists in the School of Physics and Astronomy and also in the School of Engineering to communicate the research that they do and to make sure that that research can be used to go on and benefit wider society. That can be things like having impacts on the economy or on healthcare, anything like that, and that's my normal role, is to work in an office assisting those academic colleagues. However, when COVID-19 became a threat in Scotland, the University of Glasgow announced that it was going to be a host of a Lighthouse Laboratory, which is one of the national COVID-19 testing centres in the UK. At the same time as the university announced that they were going to open and launch that centre, they also put a call out for anyone who had any relevant skill sets to come forward and to volunteer themselves to help out in the lab. I was very fortunate to have completed a PhD at the university in June of last year, and when I did my PhD research, it was in stem cell research, so I spent a lot of time working in a laboratory, and the cells that I used came from patients who had undergone hip replacement surgery. So while those patients would have seemed perfectly healthy and ready for surgery at the time that they went for it, there was no way of us knowing if the patients and the cell donors had any kind of underlying infection in their bone marrow cells. So when I worked with those samples and with those cells, I had to be very, very careful at all times that I was doing so in a really safe way, in a way that would protect all of the other scientists who I worked with from anything dangerous being airborne or present in the lab. So in order to work with those cells safely, what I did was use them in something called a safety cabinet. In a safety cabinet, there is airflow moving around all the time, so you put your hands through and you cope with your cells inside the safety cabinet, and the airflow that moves around stops anything that is potentially dangerous from escaping the hood and going into the lab. I also did a lot of different types of experiments with those cells that were quite similar to the way that the lab detects COVID-19. So I thought I had a skill set that would have been useful to the laboratories. So I volunteered myself around about sort of March time, April time, to go and work in those labs, and I was really fortunate to have been chosen. One of the best ways to explain what my role was in the lab is probably to give a bit of a general overview of the entire process of how, when carrying out COVID-19 testing, we can go from actually working at a testing centre and generating a patient sample to actually producing the results that are needed at the end. So first of all, at a testing centre, a patient would come along, or someone who wasn't feeling very well, and they would get a sample taken. So they would have a swab taken, and it would be put into a tube with a barcode on it and some medium, and that would then be their tube that was packaged along with lots of other samples from Scotland, Northern England and Northern Ireland into a van and driven all the way to the laboratory that I worked in at the Queen Elizabeth University Hospital. Once the samples had arrived there, there was then what was called the unboxing step. So thousands of samples came every day into the lab, and one of the things that we had to do first of all was some quality control work, just to make sure that the barcodes were present and we could identify whoever the samples came from, to check that there was a swab and there was also fluid inside the box, and also to check for leaks, because sometimes the patient samples could leak and that would pose a threat to the lab. So that's like the first stage. And then after that, we would take all of the samples and put them into giant plates. Now, they were called 96 well plates, and in there we would have about 90 to 93 different samples from all different people, and that format that they were scanned and racked into was noted and put into some software which would make it clear where every single person's sample was in the process. After that, there was then a step called the viral inactivation step, where a mixture of chemicals were put into these giant 96 well plates to stop the virus from being alive and posing a threat to the lab. And then after that, there was what's called the RNA purification step. And that's where we would get the important part of the virus out and make sure that it is ready for the final step, which is the testing step known as quantitative PCR. Once that's done, results would be generated. So as you can tell, this whole process has lots of different steps in it. And within the lab, people tend to be trained in one or two specialist steps. For me, those two steps that I was involved in were really the scanning and racking and the viral inactivation steps. So I spent a lot of my time collecting in samples that had already been checked for their barcode, swabs and fluid, making sure that they were racked carefully into the 96 well plates, and making sure that all of the barcodes in the correct positions were entered into software so that the person's samples could be traced the whole way through. And then I was also involved in the viral inactivation step, which was, as some people think, the most important step, because it really helps to safeguard the safety of the lab and to make sure that everything is set up for the rest of the scientists to do their job properly. I am really grateful to both the University of Glasgow and the NHS for the opportunity to go and do the secondment in the Lighthouse Lab. It was a fantastic experience where I felt I really got to use some of the skills I worked hard to learn throughout my PhD and through various internships that I've done. I met some really wonderful people. The people who work at the lab are very selfless. They're working really long hours in hot, often unpleasant conditions. So they're really wonderful people, and I'm so lucky to have had the opportunity to work with many of them. I also feel very grateful for the people who I work with in my normal job who kindly took over my work while I was away. They've done a fantastic job of keeping things afloat there, but yeah, I would just really always think that we owe so much to those staff in the labs. They're a really important part of the NHS and as part of this national effort that we're going through as a country to try and fight against COVID-19. So thank you to all of them, and I hope that they continue to keep up the good work.